Hello everyone, this is Mr. Jackson, and today we're going to be kicking off Unit 3 over genetics and heredity with Lesson 1, where we're going to specifically be looking at cell growth, cell division, and reproduction. We will know that we have mastered the necessary content of this lesson whenever we can describe some of the difficulties that a cell would face as it gets larger. So as the cell gets bigger in size, what are some of the difficulties that are going to occur? And then also we want to be able to compare what we mean by asexual and sexual reproduction, as we're going to be discussing both in relatively good detail. Now, we're going to start by acknowledging that each of us as human beings begin as only a single cell, a zygote, a fertilized egg, where a sperm will fertilize an egg. That is the first cell that creates us. And the average human body would end up containing over or nearly 40 trillion cells. So we're going to go from a single cell to 40 trillion. Considerable difference, right? And while that difference is considerable, fun fact, that actually only ends up requiring about 46 rounds of cell division. So we can go in just 46 rounds from a single cell all the way up to 40 trillion. But that number of cells is substantial, and it, kind of brings in the question, well, why do we go from a single cell with all the information needed to over 40 trillion? It's important to note cells can grow by increasing in size, but eventually most cells end up dividing after growing to a certain point. And so that's to say that the larger a cell becomes, the less efficient it's going to be in moving nutrients and waste products across its cell membrane. And it's also because the larger a cell becomes, the more demands it's actually going to place on its own DNA. And we're going to explore kind of what that means and a useful analogy as we move forward. So really, size is the issue, okay? Now, let's address some key portions here. To stay alive, a cell must allow food, oxygen, and water, all of the nutrients of life, to come in. And they're going to come in through the cell membrane. And the cell must also allow waste products to go out of the cell. So once we've used things to prevent clutter from, cluttering up the cell itself, we're going to let waste products go out through the cell membrane. The rate at which this exchange, right, so good things coming in, bad things going out, the rate at which that exchange takes place ultimately depends on the total surface area of the cell. That's just the total area of cell membrane that we have available, the amount of space where stuff can come in and leave. But the rate at which nutrients are actually used, the rate at which we create waste products, is going to depend on the cell's volume. So what's going on inside the cell? So those are two really important features to distinguish. You have the outer boundary, which basically allows how much can come in and what can go out, the rate at which they come in and out. And then you have the overall volume, the inside of the cell, which is basically like how quickly we create waste products and how quickly we use up nutrients. So as a cell gets larger, what tends to happen is that the cell membrane, the surface area, is going to increase, and the volume or those demands of the cell, what's happening on the inside, is going to increase. But the problem with that is that the surface area, how much cell membrane is there, does not increase as fast as the volume, which is all of the inner workings of the cell. So the boundaries can't typically keep up with what all the cell is doing as the cell gets bigger. And we're going to look at how that actually works. Now, here we're going to be looking at the difference between surface area and volume and how the two basically create a ratio of one to the other. And so a surface area, what we mean is just that outer covering of the object, right? The amount of surface available, the amount of cell membrane. Whereas volume is just the amount of space inside. So you can think of surface area as being the amount of space outside, volume, amount of space inside. And so for surface area, let's say we're going to use a cube as an easy example. For surface area, we're going to want to take the length and the width of a single side And then once we have that multiplied by each other, we can multiply the total number of sides of the cube, which in this case is 6. So in this case, for surface area, we would multiply 1 centimeter by 1 centimeter, and then multiply that by 6 sides, giving us 6 centimeters squared. That's our surface area for this cell. Whereas for the volume, that's going to be the length by the width by the height. So we're actually going to also multiply the height, and that's going to give us the amount of space inside the cell, which in this case, the volume is going to be only 1 centimeter cubed. Okay, so we have 6 centimeters squared of cell membrane and only 1 centimeter cubed of the overall volume in the cell. Not a bad ratio. That's to say that we have a lot more cell membrane allowing things in and out than we have actually going on inside the cell. And so we can actually compare that small cell example where all the sides are 1 by 1 centimeter. And now we can see, well, what happens to those numbers whenever we double the size of the cell or triple the size of the cell? And this is going to get a really important point across, hopefully. So if I have a cell that's just twice as large, right? For the surface area, it's going to end up being 2 times 2 times the number of sides, which is 6. That'll give us a total of 24 centimeters squared. And then the volume, since everything has become twice as large, the volume will now be length times width times height. Each one of those is 2, and so that'll end up being 8 centimeters cubed, which is just 2 times 2 to give me 4, times 2 to give me 8. Whereas if I go now to the cube that is 3 times larger than the first, now those sides are each 3 centimeters long. There's a total of 6 sides. So my total surface area or cell membrane is 54. Again, it's increasing well, but the volume is now going to be 3 on each side. You multiply that out. 3 times 3 is 9. 9 times 3 is 27. So we now have 27 centimeters cubed. Now you might be thinking, this doesn't seem like a really big disadvantage because the surface area is still way higher than the volume. And in order to understand really what's happening, we have to actually look at what's called a ratio, where we compare the size of the surface area and the overall volume to each other. And when we do so, we get a really useful breakdown, just like this one, where it breaks down exactly what we've just gone through, but I want to draw your attention to the bottom row, specifically to these 2 portions. Now, whenever we had the really small cell, 1 centimeter on all sides, that created an overall ratio of 6 centimeters in surface area to 1 centimeter of volume. So there was like a 6-to-1 ratio. I had 6 times the amount of cell membrane to serve the overall volume of the cell. But if I get a cell that's 3 times larger, all of a sudden, rather than 6-to-1, now I only have a 2-to-1 ratio. It's much smaller. So now I only have twice as much cell membrane as compared to the volume within. And as we continue to increase the overall size of the cell, what happens is that eventually the overall volume that we need to serve is greater than the amount of cell membrane or outer boundary that we have to actually serve it. And so we're going to hit these limitations, and the really easy way to remember what's happening and why we actually end up needing to divide cells before they get too large, you can think of the analogy of a city. Now, we want to limit cell growth because at some point, just like within a city, if you don't increase the size of the city, but you keep bringing more people into a city, you keep bringing more needs of people into a city, at some point you're going to hit traffic jams and traffic problems. So this is just to say that if a cell were to get too large, it would be more difficult to get the good things in and the bad things out of the cell because you're going to hit traffic, right? We're going to create a lot and use a lot faster than what we can bring it in and what we can get it out. Another problem comes in with information, right? So all of your cells have this genetic information, we call it DNA, and DNA is used to build all of the functional units within your cell, all the cell organelles, the proteins, etc. And so basically, if the town keeps growing, your DNA is kind of like a library. And if there are a whole lot of people all trying to access the same library, it's going to be really hard to get the necessary information to everyone who needs it. And so your cell dividing is kind of like basically making another acceptably sized city with its own library. So there's not a whole lot of buildup or demand placed on the same library or the same amount of DNA. And so cell division is the answer to this potential problem. So we don't want cells to get too large. At some point, if cells are not going to get too large, but we as organisms would like to keep growing, we're going to have to divide those cells. We're going to have to divide what's in them, and we're going to have to keep our surface area to volume efficient. And so the process of cell division, what we're doing is we're taking one cell and we're producing two daughter cells. And so cells are going to have to replicate their DNA, right? We have to have all the information necessary in the two new cells. So that's going to be an important part of the process. This is basically saying that each daughter cell gets a complete copy of the DNA. They get their own library. And then dividing to make more or smaller cells is what's going to keep that surface area to volume ratio high. This is going to allow for a more efficient exchange of materials through that cell membrane. So what's coming in and going out, it's a lot more efficient, and it serves all the needs of the cell. So we've covered in great detail that first, and I would say that primary learning objective, the most important part to this, but we also want to distinguish or compare and contrast asexual as opposed to sexual reproduction before we finish this first lesson. Now, whenever we refer to asexual reproduction, this is where a single parent cell, so one cell, will actually divide and produce two genetically identical cells. All that happens is we copy the DNA and we divide it equally into both cells. So the main thing to keep in mind here is that this process is going to be simple, efficient, effective. It enables populations to increase very quickly because all it requires is the cell itself. And from that one cell, we can just simply divide, make identical copies. Whereas sexual reproduction is going to involve the fusion of two parent cells. So those two cells, one from mother, one from father, are going to fuse together and create what will end up being a genetically unique daughter cell. So whereas asexual reproduction was one cell going into two, sexual reproduction is going to be two cells merging, fusing, and creating one daughter cell. So this is where offspring inherit some genetic information, some from mom, some from dad, and they make a genetically unique offspring. And it's not just wanting to identify what those two things are. We also want to be able to compare the pros and cons of both types of reproduction. These both exist on our planet, and nothing exists by accident. There must be some pros and cons to consider. Whenever we talk about asexual reproduction, you would be able to produce many offspring in a very short period of time, right? Sexual reproduction, you have fewer offspring, and growth is probably going to take more time. So that's one comparison we can make. For asexual reproduction, you don't need to find a mate. That's good if you want to do things quickly, whereas sexual reproduction might be a little more time consuming and might require a little bit more of the individual because they need to find a mate. They need to appeal to a potential mate. For asexual reproduction, stable environments, genetically identical offspring are able to thrive. But if those conditions change, the offspring might not be well adapted, so there's a risk there where if we don't have any sort of diversity or uniqueness across individuals, if one individual is not well suited and would die, that means all individuals are not well suited and would die. So we are really technically risking extinction by keeping everybody genetically identical. Whereas for sexual reproduction, in changing environments, genetic diversity is going to be beneficial. Offspring may be less well adapted to current conditions, but that doesn't mean all offspring are less well adapted. We're going to hit this point home a lot in Unit 3. We're going to hit it home as we move forward into the end of the year with evolution. But for the time being, we want to think back to our lesson 1 learning objectives. At this point, hopefully, this information has allowed us to be able to describe some of the difficulties that a cell would face if it were to just keep growing and growing and growing. Remember, this is similar to traffic problems into and out of the cell, as well as information crisis, where there's too many people trying to use the same library. And then also, as we just did on the previous slide, guys, being able to compare asexual and sexual reproduction is going to be critical before we try to build on this conversation moving forward. And so that actually will complete our first lesson of Unit 3, and I hope you found this helpful.